Do you have questions, class?
No, teacher, thank you.
Okay, so let's proceed to genetic technology. Wait for a while. Sorry. Dr. Nagla, I lose the picture on Zoom, so I will use the laptop. So there you go, class. Our topic for today has actually the same with what we have in the natural selection. But this time, class, it's a man-made. Okay? So let's proceed to this one. Can you see this one clearly? Sorry, I have to adjust, class. I have to adjust my. Okay, so can you see this, everyone?
No, teacher, there is nothing clear. Oh, okay, now yes.
Okay. So wait for a while. For genetic engineering class, I'll show you example of genetic engineering. So here you can see example of chicken, and as well as the glowing the dark fish. Okay. So how is it happen? Class, through genetic engineering class, we will form a what we call genetic modified organisms. Okay, they are already undergoing experiment. We call genetic engineering, and that's our topic today. Genetic engineering class is the process of using recombinant DNA technology to alter the genetic makeup of an organism. So for example, class, in this topic, we will know how genetic engineering helps the evolution of living organisms. Genetic engineering could not also help the manipulation of genomes of animals or plants, but also in the production of insulin. In medicine class, it could also be helpful. So as you all know, we have pancreas that produces insulin. Okay, and this insulin helps our body to prevent high level of sugar. So what will happen, class? What will happen here is that for us to produce a lot of insulin for those who have type 1 diabetes because they have problems in producing insulin, we have to use what we call genetic engineering. So number one step is to obtain the gene of interest. So how is it happen? I'll show you a video wherein it will discuss further of how we can get the gene of interest uh, to put it into bacteria rather.
Now, one of the best examples, and the one you need to know about, of genetic modification is how bacteria have been engineered genetically to produce human insulin. If you are type 1 diabetic, you cannot produce the human insulin, you have to inject it for the rest of your life. We used to use insulin from pigs, but it doesn't work as well as human insulin. Now the problem is, how do you make enough human insulin for millions of diabetics? The answer is to get the gene that makes human insulin from a person's DNA, cut it out and stick it into a bacteria. The bacteria.
You heard it right, class. Again, since there are people who have problems in producing insulin in their body, particularly in their pancreas, of course, those people we consider that either having type 1 diabetes, okay? So they need to take insulin. Okay, so where do this insulin came from? This insulin uses bacteria, okay, this bacteria will reproduce those insulin later on. So first of all, the first process is we will get the gene of interest, particularly the insulin, in a certain person, and that gene will insert to bacteria, and the bacteria will reproduce those insulin. So how is it work? So the first stage, class, let's back to this topic. The first step is to obtain the gene of interest. So this is the gene, okay? This is the gene that came from a certain person who can produce insulin. Next step is to use PCR. Okay, PCR is the polymerase chain reaction. This is an enzyme that amplify the amount of the gene of interest. Class, remember, when we say amplify, we want to have a lot of copies of gene of interest. And class, we don't need just one DNA copy. I mean, one gene of interest. We need a lot of copies of gene of interest for this experiment. Now, once we have the copy of genes, we need to find a suitable vector. So what are the suitable vector? Class, first of all, let's do the term and definition. Okay, so sorry for that. What's this format? Miss Nagla, wait for a while. Wait for a while. Madam Nagla, wait for a while. I'm already starting with the lesson.
Miss Clarice, you can use your camera on the laptop. If you want to use it. Yes, it's okay, Miss Clarice. Start.
Wait, I'm getting confused now. Class, can you see this?
Yes.
Okay, so there you go. One of the examples of vector is plasmid, class. Vectors can either be plasmid, viruses, or bacteria. So what are the plasmid? Plasmid is actually a bacteria, but these are the bacteria who have a ring of DNA. Okay? So these vectors used to transfer the genetic material such as DNA sequences from the donor organism to the target cell of the recipient. So as you look at this one, class, this vector, which is from the bacteria, needs to be connect to gene of interest, which is the genes for the insulin. Now, In this topic class, we will use what we call restriction enzyme. Okay? This restriction enzyme, if you're going to remember our topic about the replication class, okay? Restriction enzyme, also called restriction endonuclease, these are the enzyme. A protein produced by bacteria that cleaves DNA at specific sites along the molecule. So for example, in this picture, you will see an example of a restriction enzyme where enzyme cuts the DNA. The DNA from a gene of interest. Okay? So once it cuts, look at this one, we have a new fragments here that have single-stranded overhangs. Okay, why there is overhangs? Because class, this fragment we will connect it to a vector, vector which is the bacteria. Okay, so this is restriction enzyme. It cuts the vector, and then the next procedure will be the DNA ligation. DNA ligation class, it happens where you know that ligase is like a paste to seals the gap. So look at what happened here. So the vector class is actually sealed with the gene of interest through the use of DNA ligase. Next process is the insert a marker gene. Marker gene have also different term called gene coding. It is for easily identifiable substances that can be used as markers. So class, this is the ligase that uses ATP or the energy. Look at this one. We have two genes came from two different species. For example, the bacteria and the human genes. Now, once they are already attached to each other through the process of ligation, it will become a recombinant DNA. Recombining the DNA from the gene of interest and the DNA from the bacteria. So let's proceed to genes coding. So I'll show you here example of a gene coding. So as you look at this one, since we use bacteria class, okay, I have already explained what's happening to this one. We get the gene to clone or the gene of interest. Now we need to paste it into a plasmid. Plasmid is the vector class. So as you look at this one, we use ligase to paste it. Now, we need what we call gene marker. Gene marker class, because this is our DNA. This is our DNA. Okay? For us to insert this to bacteria, because class, I told you earlier that insulin is being used for the production, I'm sorry, the bacteria is being used for the reproduction of insulin. So this rDNA needs to insert into bacteria. But how will we do that? The scientists use a solution, okay? A solution wherein there are a lot of bacteria inside, and as well as a lot of nutrients, okay? Now, this rDNA, okay, this rDNA will actually place inside the solution for the bacteria to take it. Okay? But class, there are times that the bacteria will not get the rDNA or the recombinant DNA. Okay? So, how we will know if the bacteria already took our rDNA? So here it is. Remember the gene coding or the marker we put here. This is actually the antibiotic resistant. Okay, this is the antibiotic resistance. Once we place the bacteria here in, for example, this is our Petri dish. once we place the bacteria there with nutrients, and we want to find out which bacteria already took the rDNA, we need to put antibiotic as well in the solution. Okay? Now, those bacteria who did not take the rDNA, who's having antibiotic resistant, will actually die. Okay? Because the rDNA is having a gene marker rather. I'm fixing my videos. So there you go. It's having a marker or the as antibiotic resistant. That's actually been helpful if we will put antibiotic inside. Then those who take the rDNA will actually live. And those who take the rDNA, those are the bacteria we need for the production of insulin in human body. So class, is this clear or there are confusion? I will ask questions. Are there any confusion, class?
Teacher,
Yes?
can you explain again the when inserting the DNA into a plasmid vector?
Okay, sure. So, wait for a while. Sorry, I'm using 2 laptops and 1 cell phone as well. I'm getting confused now. Alright, so this one class, this is a plasmid. I told you before, it is a bacteria which has a ring of DNA. Now, it has a gene of interest, which is the insulin, a gene for insulin. And the reason why they are already pasted to each other is because of the DNA ligase, okay? The DNA ligase is the one that sealed the gene of interest and the plasmid. Now, class, we need to use a marker. Since this is a bacteria, this is a gene of bacteria, we put marker for antibiotic resistant. When we say resistant, meaning to say these are bacteria that could survive in antibiotic. Okay? Now, for example, these are DNA class. We need to insert it again to the bacteria. Okay? Really having. Wait for a while. Okay? So class, for us to find out if what bacteria already took the rDNA, we need to place them in the solution. Okay? So this is the solution. Okay, this is the solution. This solution is have nutrients for the bacteria to live. Okay, so there is solution inside as well. Not only the solution but also bacteria as well. Now this rDNA, we will place it here inside. Okay, because we want the bacteria to take the rDNA. Okay. Now, once the rDNA is already inside the solution, we want now to know which bacteria already take the rDNA. So for us to find out if which bacteria already took the rDNA, we use, for example, this one, a Petri dish, wherein there are nutrients inside. Okay? Now, the bacteria will live if there is nutrients. But class, when we put antibiotic class, this is different. They will die because it's antibiotic eh. Okay? But class, those bacteria who take the rDNA will not die because there is a gene marker we call antibiotic resistant. Okay? Now, once those bacteria already live because they have antibiotic resistant, we will now incubate this bacteria to reproduce. And of course, if they will reproduce, the insulin that they are having will actually reproduce, like millions of copies of insulin. And this is what those diabetic person will take once they need it for their for the insulin intake. Are there any confusion? I'm confused with my cell phone. Sorry. Don't think this is good. Lamia?
Yes.
Lamia. Okay, question.
Nothing.
My question is, can you tell me what is the gene of interest in the step by step of genetic engineering?
Repeat.
What is the gene of interest we get from a person? From what I've discussed,
Okay, wait a second. Genome. Teacher.
Sorry.
Teacher.
Yes?
I'm confused.
Which part?
Repeat your question. I didn't get it.
Okay. Nadine? Wait for a while, Lamia, ha? Nadine?
Yes,
Nadine?
teacher.
Okay. Can you tell me what is the gene of interest?
Mm, the gene that you would put in the plasmid?
Yes. But what particular gene we are taking?
There's not the restriction enzyme. I forgot, teacher.
Okay. The gene of interest that we need to take from a person is insulin. Okay? Okay, I will just repeat this step 1. Okay. For the step 1 class, remember there are people
You
who
send it to
have
us. We'll study them and we will be able to answer now because just now we studied it.
I'm sorry?
If you send us the PDF which you are doing it from it, we will be able to study it.
Please check your Google Classroom. It's already there.
Okay. Just we will
Okay.
study it, then we will answer your question if it is possible.
Okay. That's good. That's good to know. Anyways, class, for the finalization of this explanation, I want you to know that the genetic engineering is very helpful in terms of medicinal purposes. For example, the production of insulin. Because class, we already tackled in hormones class the production of insulin in the pancreas. There are people who's having a hard time in producing their own insulin in their pancreas because they have type 1 diabetes. So the scientists, what the scientists did is they produce insulin using bacteria. Okay? So class, in the bacteria class, we will merge the insulin gene, particularly the gene of insulin, into the DNA of bacteria. Okay? So once it happens, this is what we're going to call as rDNA or recombinant DNA, wherein there is a recombination of the DNA for the insulin and the DNA for the bacteria as well. So I hope these steps are clear. Last question for Wail. Wail?
Yes, teacher.
You asked me about the how the rDNA will insert to the solution. Can you tell me what did you understand with my explanation about the insertion?
First, that ring of DNA, right?
Okay.
Okay, the ring of DNA, we will put it in the solution. The solution will have the bacteria, and we are about to know which bacteria will take the ring of DNA. So to know which, we will put it on that what do you call it, the dish.
Petri dish, yes.
Okay, and in the dish we'll have also the nutrients,
Mm-hmm.
and the bacteria will live on the nutrients,
Okay.
and the others will die
Why do you think the others will die?
because there is antibiotic resistance.
Okay. And
Yeah,
why there is a bacteria who make it live in the solution?
because of the nutrients.
Aside from the nutrients.
Um, no, I forgot that part.
Okay, remember that pink one? Remember this one?
The
There
antibiotic
is a
resistant.
process of what we call gene code, gene marker. Okay, gene marker is actually what we call antibiotic resistant. Okay. Well, very good. You explain it well. But for in the last explanation, the reason why there are bacteria who can actually live in the solution with antibiotic, it's because they have antibiotic resistant. Okay? Imagine this is antibiotic. And when we place it here, which having a lot of bacteria, of course, the bacteria will die. Okay? But then, since there are bacteria who take the rDNA, which is having antibiotic resistant, they will live. Okay? And then that's the time there would be a lot of reproduction of insulin. Okay? Is it clear now?
Yes, thank you.
All right, thank you. Okay, if you do not have any questions, please check the Google Classroom. I put all the notes there, including the videos. This is a very good video class. I also included it there. So please check it. Next time, we will be having your I think third worksheet. Okay? So do you still have any question? All right. Thank you so much, grade 12.
Bye, teacher.
Welcome.
Bye.
stop.